A little bit about DPX. I don't want to go through the entire timeline, but just to give you guys a quick synopsis of the history of DPX. Problem solving is what DPX is all about. And in fact, the vision is to empower scientists by offering versatile products that can integrate with any workflow or method. Dr. Bill Brewer, the founder of DPX, first conceptualized this product line, the dispersive pipetting design, back over 20 years ago, working in a lab for South Carolina law enforcement department. He's a crafty person, and the wheels are always turning, and he just was looking to build a better mousetrap, working in a toxicology lab, and essentially came up with this idea for a loose resin inside of a tip bound by two barriers that would actually freely disperse in your matrix of choice when you're aspirating and dispensing, whether it be on a handheld device or an automation system. And that's pretty much how DPX was born. A few years ago, he patented the tip-on-tip technology, and that's what I'm going to focus on today. But more recently, and really exciting too, are some other products that are opening the doors to new avenues for us in the world of proteomics and genomics, nucleic acid extraction, both DNA and RNA. So it's really exciting times at DPX, and I'm glad you guys can all join to be a part of it. So the agenda today, like I said, I'll be speaking mostly on the tip-on-tip, the proprietary and patented technology that is extremely simple. I often wonder why, with my years of experience, that I never thought of doing this. So that's kind of kudos to Dr. Bill Brewer for coming up with this. It's just so simple and easy. So you'll see a few methods that I'll talk about, how we automate them. You can even use it by hand, but from simple filtration workflows to a cleanup and even solid phase extraction using the tip-on-tip with some creative tools and designs. And then you'll be able to meet the team. I'll just do pictures and introductions and so forth. It's a little more information on how to order, and then open up for questions. So just looking at this slide, this actually kind of shows in chronological order from left to right the development of the product lines for sample preparation. I'm going to use my mouse. I think you guys can all see the mouse. So starting over on the left, this was the main focus of the talk from last week. It's our INTip solid phase extraction, basically dispersive pipetting technology. Bill, like I said, Bill Kemnitzer had done that presentation last week. If you missed it, I believe that we now have it just posted. Might be on the website. I saw it on LinkedIn today. The tip-on-tip filtration. You can actually see very simply, it's actually a tip that is on a tip. And so I'll talk more about the anatomy and design and methodology. The latest and greatest. It's such a cool picture. I wanted to have it on the slide here. So this is our INTip size exclusion tips. They're shipped actually with a dry resin, so there's no mess in shipping. The automation, or if you're doing it by hand, you actually swell, add your sample, and your sample typical proteins or peptides will then fractionate. If you have questions or want more information about that, feel free to send us an email or give us a call. General anatomy for the tip-on-tip, as I mentioned before, not a very creative terminology. It is exactly that. It's a tip on a tip. Now, the real beauty in that is that the bottom tip that has the filters can fit on any tip, or just about any tip. I don't want to say any tip because somebody might challenge someone to go find a tip that it can't be done on. But what I mean by that is that really any automation, any handheld pipette or electronic device has usually a special tip. The bore size up top fits on that particular automation or pipetter, and sometimes is unique to that particular instrumentation. So that top tip we don't touch. We just leave alone. It's better if it can be wide bore for certain applications. If it's a real viscous, if we're doing maybe a protein crash or something like that, I'll show you in a couple slides. But the bottom tip has a gasket that will seal onto that tip. And I'll show you a quick little slide, a video in the next slide showing exactly how that does that. Some of the things that this can now do, again, because it's such a simple design, is replace centrifugation. One of the hardest things to do, and like Carmen said, I have years of experience in the automation world, and probably one of the hardest things to do is automate centrifuge. It's just really not done. We've tried it. I know several people have tried it. It becomes the Achilles heel in any type of automation system. So having an alternative in something as simple as this that is extremely amenable to automation it was really exciting to me and to a lot of customers that I know. It'll filter down to less than a micron, no positive pressure or vacuum devices. So really no extra hardware other than just a pipetter is needed. And what that results in is just a low cost and extremely efficient design for filtration. Extending the life of the LC column. So it's, I say that because a lot of workflows, people are doing dilute and shoots, and probably in just about every dilute and shoot application that I've talked to customers, they have a centrifuge. So it's probably easier to say dilute and shoot than to say centrifuge, aliquot, dilute and shoot, right? So just about everybody is using a centrifuge. So this will, again, eliminate the need for those extra accessories. Quick video, you'll see a robot with the top tip that is standard just come down and simply with really no force, just press on those bottom tips. The methods that we have come up with can be, as I mentioned, simple filtration all the way to total solid phase extraction done with a tip on tip, or a cleanup. Cleanup and solid phase extraction go hand in hand. It just depends on exactly what you're trying to do. Cleanup is more of a bind in a loop because you're binding your interference, where the solid phase extraction, you're trying to bind your analytes of interest and then wash off the interferences. A little bit longer of a process. Cleanup is just a quicker and easier, but it really depends on what your workflow is. Filtration has opened the doors for a bunch of things, including protein crash. So again, working from a variety of different matrices, whole blood protein precipitation is a very common one. We've also, in the tox world, after like I said, a typical dilute and shoot, after a urine hydrolysis for maybe a drug of abuse panel, we can do a simple filtration that'll get rid of proteins for if they're sick patients, but also the beta-glucuronidase, the enzyme responsible for hydrolyzing the urine. So anything that might junk up your column, your LCMS, we try to get rid of, and this is a simple way of actually doing it. So with that simple filtration, we have even a variety of different tips that we can put on there. Our team that you'll see towards the end of the presentation, most of them are PhDs, have years of bench science work. And it's the real fun thing, I think, for any scientist is the actual lab work. It's like the actual maybe R&D if you're an R&D person, but we're partly a custom shop as well. So, we have several workflows that we've done, we've automated, we work with customers on papers and so forth. So there's a lot of turnkey type applications that we have, but we also welcome the challenges, things that, what are the real challenges in your lab that you're struggling with? Will challenge us on that. Let us try to see if we have a solution or can even design a solution or tweak a product to meet your need. Here we're showing two separate tips that can be on the bottom of that for that filter tip. This is our dual phase filtration tip. So it provides a maximum filtration without clogging. Very common in the cannabis world, which has become more and more common now with especially a lot of states now becoming and improving the recreational cannabis. The low porosity filtration tip is probably the most common. It's because we use that with probably a real dynamic range of matrices from urine to blood to cannabis as well. But there's also others that offer lower detection level limits. They're used for what we call ultra pure grade filtration tips and so forth. So And I'll have to apologize here to Carmen because I couldn't find the video with the new branding. She's done such a fantastic job on the new branding. So she's probably cringing at this, the old branding, but I promise you, Carmen, I'll make it up to you in one of the next videos. So here we just grab a 1 mil tip, a standard tip. It's a wide bore to prevent any clogging with the particular instrument that it's on. And you can see it mixing the acetonitrile with the blood a few times and getting a really good crash. You can see that nice gooey mess there. So now it'll go pick up some fresh tips that are even conductive too. So if you were going into a larger vessel, if you had more volume in there, you don't want it to overflow into the adjacent wells. It'll use the liquid detection capability of the robot. Then it'll pick up that low porosity bottom filter tip that I was just talking about, and then just simply dispense through. And so you'll see the precipitate will be stuck on the filter and a clean filtrate being passed through. And there's a good close-up shot of the precipitate and a nice clean filtrate going through. So again, I mean, really, you can see that was real time, how fast and simple, straightforward that is. No vacuum box, no centrifuge, etc. I was talking earlier about a dilute and shoot. I don't know if we've coined this. I kind of like the filter and shoot. It's one step, I guess, better than, I'll say, quite biased, but better than a dilute and shoot, simply because, again, no need for a centrifuge. It can be completely automated, with a robot, allowing you for full walk away time. So if you have, depending on what robot you have, you can do, in this case, what we're showing about a beta-glucuronidase. This is a urine hydrolysis for a pain management workflow. So people could actually load the machine with their urine tubes and just completely walk away, and the machine will hydrolyze, and then it'll pick up the filter and do the filtration into a clean plate or tube, and that can then be taken right over to your LCMS. Carmen mentioned she was going to actually put a poll in there, so that was nice timing right there. So she's asking, what is your method choice for urine drug analysis in the SPE dilute and shoot? So that probably popped on everybody's screen. Can I write in my own box, Carmen, and say filter and shoot? So
no, you can't write in your own. There's about I'll leave it up for a couple more seconds and then I'll take it down. But you can keep
Okay, I'll just continue. People can multitask, I guess. So this is a picture here of your hydrolyzed urine sample. In this process, we just added a 1-to-1, 1 part methanol to the solution, and then did the bottom filtration technique that we just showed. You can see the recoveries are all, I'll say perfect. But what's more impressive is the consistency. So this is a 96 well plate up here, and we started with 200 microliters, dispensed that through the bottom filter. These are the resulting volumes. So you can see after, a certain amount was trapped onto the filter and didn't pass through, this represents a coefficient of variation less than 2%. So a very consistent way, and there's actually papers out that have proved that a filter and shoot, as I call it, is much more consistent and better than the traditional dilute and shoot methods. As I mentioned before, too, another really popular, it's growing in population over the course of the last 6 months to a year or so, is the cannabis testing. No matter what your downstream test is, whether it's just simple potency testing or maybe it's metals or pesticides, mycotoxins, or whatever it may be, the first step in all these is to remove particulate. So whether it's the flour, the oil, the edible, everything needs to be homogenized and filtered. And the way that people have been doing it, it's just painstakingly slow and manual. So offering this simple filtration, especially on a robot, as your sample throughput increases, has just been a blessing. I've heard it's been called to these labs. So now being able to just again walk away from that initial process has been really impressive. It's fast and simple, and when you automate it, it's really easily automated, and the cost, it's extremely inexpensive compared to the ways of doing it. And any of you that work in this lab or are familiar with these, I mean, that's pretty much the goal of almost any of these labs anyway, is to try to get the costs down and results still looking good. So moving on from the simple filtration now to talk quickly about the cleanup and the solid phase extraction if you're using the tip-on-tip. So the cleanup, again, all really depends on what you're trying to do and what your workflow is. It's compatible with a variety of different chemistries and works with, again, a bunch of different matrices, no matter what the viscosities are. But the real beauty in doing something like this now is, instead of buying tips that have already come prepackaged with a certain amount of resin in it, you can provide your own chemistry, and keep it on the deck. You can buy it in bulk and sort of do your own R&D right on the deck. And I'm going to show you in a few slides a customer that we actually work with. There's been a handful of them, but one in particular that asked us to provide well plates with resin in it, so that he could or they could actually do the solid phase extraction with the tip-on-tip and really customize it to their exact need. So in this particular case, so you'll have now you add your sample to the sorbent or vice versa, mix that up. Then you'll pick up that bottom tip, and you'll dispense. So the resin inside will bind the interference and pass your cleaner sample into a new tube or plate. I have a video coming up in a couple slides that'll actually show the tip-on-tip SPE, which is a pretty cool video. So this tip-on-tip SPE has really opened the door now to many more possibilities, as I mentioned really early in the presentation. Getting us into some proteomics applications like protein purification, peptide purification. More exciting to me anyway is the genomic side of things, so nucleic acid isolation and purification. And what this slide is showing, if I start up top here, it's showing a magnetic bead application, which is pretty much the standard in nucleic acid isolation. And it works, but the mag beads are pretty expensive. But people have all gravitated to this technology over the years. So you would add your sample to a solution of beads, the magnetic beads, mix it all up, and now you would have to have your robot or manually move the plate over to where your magnetic, usually they're post if you're working with 96 well plates. And the magnet then will attract the beads that have hopefully captured your analytes of interest. And now the pipetter can come in and actually remove the supernatant and then do a wash. It'll move it off the magnet to add your wash solution, to mix it up several times, then move it back over to the magnet and remove the supernatant again. And it repeats this several times. So you can see how slow, because you have to manipulate the microplate, moving it back and forth off the magnet. Then when you're ready to elute, you do the same thing again, bring it over to the magnet, before the magnet, add your elution solvent, bring it to the magnet so only the beads now get attracted, and then you transfer your supernatant with your isolated analyte. The DPX method for doing the tip on the SPE, again, you don't need the magnets. Here, this is showing, this could be a proteomics example. So now you would be adding, to the deck, you have your sample, and could be your resin of choice. In this case, we have a protein A, protein G, which is pretty common when you're working with a protein purification or peptide purification. And you would then aspirate and dispense to mix it up really good and get the appropriate binding. Now this particular workflow, I've seen this where this, I'll call it incubation time or residence time, could take anywhere from 15-20 minutes to overnight. I've had people tell me that they do an incubation at 4 degrees overnight. Some other people like doing a 37-degree incubation, which you don't want to tie up your robot for that long of a time. So being able to do it this way, so this, this mixture could be incubating offline. It could be on the deck if you're not using the robot, but you could have it on the bench somewhere, or at whatever temperature that you need it to be for as long as you need it to be. And then when that residence time is complete, then you can bring it back over to the automation. And now you can simply aspirate, and then you'll pick up that bottom tip again, and then dispense. And now you've trapped if it's an antibody or what you're looking at, you've trapped that down into the tip. And now you have this tip on tip that can actually be treated like a simple dispersive standard tip, meaning you can now move it over and you can wash as many times as you need it to, and then move it over to your final plate and just hit it with an elution solvent to cleave your antibodies. Really clever. And again, using that same tip-on-tip technology to do it. A couple of the differences here is that you have choices again for that bottom tip. So you could have that disperser, more common for the SPE when you need to actually disperse that resin inside that tip. But it's a single phase filtration tip. And the Fritz can be different size depending on what the resin size is. We have a variety of different Fritz sizes for those. And let's show a quick video here that actually does a really nice depiction of the tip on tip for solid phase extraction. So as this site it starts, we're only using one tip just to show. It's actually, I think it was done better with just one tip to really help you focus exactly what's going on. I should have paused it there. You might have seen the resin that was inside that well. So we're adding our sample to the resin, and I'm gonna stop right here for a second and notice. Well, I'm running short on time, so I'm gonna go. You'll see that nice green color. I usually like to quiz people and say, remember when you were a kid, you had crayons and you didn't have the green one. How'd you make that green color? You had to actually color first with yellow and then color with blue. Well, this is exactly what's going on here. So we started with our sample solution that was green, and our interferences that we're trying to wash off now are represented in the yellow color, and our final analytes that we're trying to detect are the blue color. So yeah, I just think this is a really cool video. You can see it washing off right now, getting more of that residual yellow color, out from the sample, and giving you a nice clean, clear blue color here as it's dispensing into the elution solvent. So again, just real simple, real neat. Carmen, I love the video. I told you I'd make it up to you with the new branding too. So this was such a really neat video. And here you see everything. I'll pause here. You see the gasket forming the nice seal here, the disperser that I mentioned, your customized sorbent that's here, and then your single phase frit at the bottom. Again, fast, easy, customizable, just really simple. I mentioned earlier about a customer that we helped out by supplying plates with resin. And so here we have the ability to dispense and send the resin inside of a microplate instead of a tip. This was an option that he wanted. And the main reason was because he loved the idea of the tip-on-tip SPE, because his wash volumes were he had so many washes with so much volume. I think, if I remember correctly, it was about 3 different wash solutions, and he needed 5 mils of each solution to wash. So it was just taking forever with any technique that he tried. And so again, this is that creativity that our team has. And after a couple conference calls with the customer, came up with this vacuum block. Now the idea here is, if you take the videos and everything I've explained about the tip on tip, we'd first drop the bottom tip off onto this vacuum block. And then on the deck, you'll have this resin plate, you'll have your sample. You can do as much mixing and incubating as you need to. And then you'll aspirate with the top tip out of your sample and dispense it into your filtration tip. And now your sample is bound to the resin inside this tip. And now you can come over with a 1 mil tip and you can just cycle through as many washes as you want. In this case, he went over to, wash 1, he added the mill, added the mill, added the mill. And, I did that, 5 or 6 times. Then he went to his wash 2 and did the same thing. And it was just super fast. He got this process down. What was really exciting for him, with data that was even better, he was close, but actually, in his words, it was better. But he took an hour and a half process, I think he said it was, down to about 10 minutes. So yeah, he was ecstatic about that. Kind of wrapping it up here, I just wanted to talk quickly about what DPX can do. If it's something that is already done and you have automation, we have package files and scripts already written on several different automation platforms. It can be as simple as sending them and tweaking them virtually, whether it be through TeamViewer, or we're more than happy to come on site and help you with this. Typically, if you're a new customer and you have something that you want to try in terms of maybe a proof of concept, we'll send you a tip for a manual pipetter, whether it be a single or electronic multi-pipetter, and you can try it out by hand. And if things look good, and then the next step for you is you want to automate it, we can come on site from there. We'll provide you with the appropriate tips in the scripts and actually help you get you going. It's something that we do at no extra charge. I know a lot of these companies now, they charge an arm and a leg for app support, especially after this whole pandemic that we had. Resources are slim. So in order to get somebody to your lab, boy, I can't even imagine. Well, I know what they charge. I don't feel comfortable telling anybody, but they charge an arm and a leg, basically. So this is something that's a service that DPX does. And again, just to get you using our tips, it's something that will help you do. So here, real quickly, you can see again a variety of different matrices, and these are all things that we have scripts for, already. So hopefully you see something that maybe sparks some interest. And again, you can certainly reach out to us for some help. So wrapping it up here, just a couple last minute bullet points here on the filtration tip. So hopefully you're able to see, the simplicity of it, the efficiency of it, being able to replace, 96 well filter plates, for example, and getting rid of any type of auxiliary equipment like the centrifuges and vacuum boxes, positive pressure devices, and so forth. One of the real things that I think is a huge plus is that each tip is separate. What I forgot to mention on that vacuum box that we designed is that each tip is isolated from its neighbor, so there's zero chance of any contamination when you're pulling vacuum, splashing from one tip to the other. And that's the real advantage, I think, of using these tips instead of for example, a 96 well filter plate. I've seen, whether using a positive pressure device or a vacuum, what happens if one of those wells gets clogged. I've seen it just overflow into the next and just contaminate the adjacent wells. By using individual tips like this, it doesn't matter if one of those tips, if you have a really, really bad sample, a real sick patient, and it's just like trying to pipette mud, only that tip fails. The adjacent ones are not affected because they're all individual. That's one of the real beauties, in my opinion. So, hopefully you liked what you saw. I will ask you to, challenge us, contact us, see if the tip on tip works for your workflow, or certainly ask us questions. We're more than happy to help you. So quickly, our team, the first founder and CSO, Bill Brewer. I said Dr. Bill Brewer, and he's still a hands-on guy. He is the happiest when he's in the lab. And those of you don't know me are probably chuckling right now, because he doesn't like the business side of being a business owner. He just loves being in the lab, playing and creating. And again, I think that's why DPX is so successful. And also too, because of the rest of the team. Dr. Kaylee Mastriani, she's our director of operations. And again, a PhD background, and she does a lot with the application side of things and method development. And it's a pleasure when talking to customers. And those of you, I think, that have talked with her can share that sentiment. Dr. Bill Kemenser, he's my counterpart on the West Coast. Again, we're hands-on guys, not just, sales. I never liked saying not just sales. I think there's a lot more to it than that, but, we're customer centric, and, customer comes first, and Bill does a lot with method development as well, in the application support on the science side. Nick Chestera, application science, tons of experience in clinical diagnostic. He's kind of our mass spec go-to guy. And he's been a great addition to the team. Dr. Matt Fitz, our molecular biologist, we hired, I think it's been a year. He's going to kill me for not remembering, but yeah, he's buried in the lab. We give him, we tell him what to do, and he's over there developing new methods and products for us, getting us into the proteomics and the genomics world. So he's been doing a fantastic job. And then our lovely Carmen Adamson, who spoke earlier and did such a nice introduction for me. She's been fantastic. That's why you probably heard me mention a couple of things about the brand. She's the brand manager. Bill Brewer absolutely loves the design, as we all do. She's done a fantastic job. She provides support to the sales team as well as account management, and she's been great. Bottom right here is just sales. That pretty much goes to all of us. If you have questions or anything that maybe I didn't cover or that you want to learn more of, you can certainly reach out on the sales@dpxlabs.com. And again, I just kind of love this whole challenge. Yes, that's what we love. You get something that that is a challenge in your lab, send it our way. Yeah, we're more than happy to help you. Real quickly now, we're down two. We got two left in April. So the 1 o'clock on Tuesdays. So April 20th and the 27th, you'll see the Sally coming up next week, and then the INTip technology for improving throughput and efficiencies would be the week after that. And so with that, I'll open it up to questions. Sorry, Carmen, I kind of went over, but I was trying.
That's okay. Yeah, so we can take any questions now. I know we kind of hit all the way to the end of time. We're trying to keep these under an hour, but we're happy to answer any questions that you guys may have about this tip-on-tip technology. Like John said, it's really simple, but it can cover a broad range of sample preparation applications. So it's really unique and cool and just open to a lot of possibilities with this technology. And you can type questions in the chat, or if you're not shy, you can unmute yourself and just shoot them at us.
I'm going to click on.
Hi, John. I have a question. Can you hear me?
Oh, yes, sure.
Thank you for the talk. My question is, will these tips work on all types of automated liquid handling systems? And can you please provide an example?
Sure. Yeah. So, yeah, I think the real exciting and part of that tip is that because it is being attached and held on with just a simple gasket that provides a really nice seal, you can pretty much use almost any automation, from a handheld pipette, electronic pipette, to virtually almost any automation out there. There's some limitations. Really, it's usually software. So because it's not the tip itself that's being picked up by the mechanics, it's just the tip being picked up by a tip. So if the software is able to handle that, and there's some that, like, for example, probably the two most common, Hamilton, you saw probably a couple of videos where I was showing Hamilton, the Nimbus platform, as well as the Star platform. They have built into their software where you can pick up the bottom tip and then adjust the overall length of that tip. So that has worked well. TCAN has some relative moves that you can do after you pick up the bottom tip and tell it to, don't go to the bottom of a plate, but you go a certain distance above that plate to make up for that extra length. So yeah, it's I haven't played with all of them, so I don't know which ones can and can't, but chances are we can get it to work.
We also have an application note from a toxicology lab. I believe it was Orange County Crime Lab, I want to say. They presented a poster on using tip-on-tip for the protein crash for the blood, and then they have a DPX method for some of their further downstream extraction protocol for a variety of other matrices besides blood. But for the blood, they start with the tip-on-tip on the Integra and then go to a DPX extraction.
Thanks, Carmen. Thanks for the question.
Thank you.
Anyone else? I know people get shy and don't want to ask questions, but you have our email. Certainly contact us offline, and be more than happy to talk to you and help you any way we can.
And I know we have a lot of tox labs that joined us for a webinar. And if you do a lot of blood work, that last webinar that John mentioned, we're talking specifically about blood and serum matrices and the variety of applications that we have with that, including tip-on-tip and SPE methods. Well, we'll wrap it up here since we're kind of getting close to the hour. If anyone wants to, how soon could we get some samples for the protein crash in blood? We keep the filtration tips in stock, pretty much all of them. So depending on the time of day that you email us, we could possibly ship a sample that same day. And so those are a really quick turnaround time with those filtration tips. And for the protein crash specifically, that low porosity filtration tip, if you email me this afternoon and I can get your contact information, I can get you some samples out pretty quickly. That was a question from the chat, by the way.
Yeah, and I know who she is, so I'll
Okay.
reach out to her. So thank you for that question.
All righty. Well, we will wrap it up. Thanks again, everyone, for joining us. Again, we recorded this webinar, and we will get the link to the recording out to everyone.
Great. Thanks, Carmen, and thanks everyone for joining.